In October 2010, about 600 scientists from around the world gathered in Indianapolis, Indiana, for the 7th International Conference on Frontotemporal Dementias. 5 of those scientists took time to chat with me about the state of research in their field and the progress that has been made in recent years.
4 years ago, it was a very exciting meeting at that time because they first had the identification of mutations in progranulin as the gene for tau-negative and ubiquitin-positive frontotemporal dementia. And at that same meeting, they had also found that TDP-43 was the protein that was actually in those inclusions. And those were 2 really very important discoveries. And now for the first time, after 4 years, people have presented their mouse models and like zebrafish, Drosophila, different model organisms. And they've started to tease out the function of these proteins, and I think that's very interesting, and hopefully now we can start doing very specific studies and hopefully find some new targets for therapies using these models.
Frontotemporal dementia research is progressing very quickly. We actually have got to the stage where there's a number of pathogenic proteins that have been identified. How they actually interact over time is being identified. And all of these are progressing to a stage where there's excitement because potential treatments are very close at hand. So having this conference where you get everybody together to move the field forward is actually extremely important.
I think the excitement in our field is really palpable. We can see the possibility of rational therapies. Increasingly, I think we understand that there are different molecules responsible for different subtypes of frontotemporal dementia. Some of the molecules may be very pliable from the scientific perspective. And I believe very strongly that the progranulin mutations may be very treatable in the next few years.
What are some of the current challenges in this field?
We urgently need biomarkers and for FTLD, and so that we can distinguish between the 3 major categories, tau, TDP, and FUS. And the reason why that is so important is that clinically they look the same. And again, treatment for these category of diseases are going to be different because they're different molecules, different disease proteins, and they have different disease pathogenesis. And so it's crucial to really come up with biomarkers, either imaging biomarkers or biochemical biomarkers, that we can distinguish. And when the patients are still alive, whether they have one type of pathology versus the other.
We continue to look very hard for the chromosome 9 linked gene associated with frontotemporal dementia and/or ALS. That gene has not been identified, and this was a topic of discussion for a good part of the meeting. What is this gene? What does the gene encode for? And how does that protein fit into the complex TDP-43 progranulin FUS pathophysiology?
What lies ahead for frontotemporal dementia research?
There's an incredible interplay between basic science and clinical science, and I think we clinicians are moving toward an approach for clinical trials that will involve biomarkers, neuroimaging, help us to produce rational scientific trials with small numbers of subjects that will tell us a large amount of information.
I think that we'll actually be able to know when to treat by earlier identification of patients. And I think there's a great willingness in the patients that people treat to actually participate in these new exciting studies. So I think ahead is only good news.
Therapeutic trials in progressive supranuclear palsy have begun, and this is going to spill over very nicely into the FTLD field. We very likely will have at least a couple agents being tested in clinical trials over the next couple of years.
I think there's just a lot going on in the field, both FTD and ALS right now, and I don't think I can predict what's going to come in the next meeting because it's going to be probably something completely novel again.
I think that there's going to be really an explosion of animal models and also mechanism of disease, and I think that both FUS and TDP in particular, they define really a new class of molecules, and that we really do need to understand the biology, for example, and what are the, functionally, what do they do? What are their RNA targets and DNA targets? By identifying those targets, RNA and DNA binding targets, and by understanding the disease mechanisms, hopefully we will come up with targets for therapy. The key is that eventually we've got to cure the disease.